Guess what time it is? It's time to talk. Just kidding. It is time to go over how to fix your score on the 422 FRQ on photosynthesis if you are choosing to do so. So let's look at question one. Question one says fossils of a microscopic organisms are found in rocks determined to be over 3.5 billion years old. Identify two pieces of evidence that would help answer questions whether these organisms were photosynthetic. Well, the first thing they would need to be photosynthetic would be probably, first thing that goes to my head would be photosynthetic membrane or chloroplast. Okay, so a chloroplast would be a number one thing they would need to be photosynthetic, right? At least if they did not have a chloroplast, maybe some photosynthetic pigments, right? And I'm not looking for you to say photosynthetic pigments. How about photosystem 1 or photosystem 2 or chlorophyll, right? If I see words like photosynthetic pigments in your answer, that means that you went straight to the scoring guidelines and wrote what was here. Okay, so I'd like to see that you actually know what these pigments are, that they're chlorophyll. Okay, all right, another one. The atmosphere at the time. If we had photosynthetic organisms, the atmosphere probably contained oxygen. So high levels of oxygen gas in the surrounding rock would be one of the high level indicators that we have a photosynthetic thing. Now, we can't say cell wall because cell walls are also around bacteria, right? So the cell wall, although it is around most plants, it wouldn't be around, it'd be around bacteria even if they weren't photosynthetic, right? Okay. So another thing could be molecules being present that are associated with photosynthesis, right? So if we think of photosynthesis, what kind of molecules do we think of? NADPH, ATP, high levels of glucose, so things around it that are involved in um, photosynthesis, right? Okay. All right, so let's look at question two. Now, this one's a little tricky. A controlled experiment was conducted to analyze the effects of darkness and boiling on the photosynthetic rate of chloroplast suspensions. That means we had the chlorophyll pigment there, and it was in a suspension, meaning it could still do photosynthesis, right? So it was suspended in a solution, and then they were going to look for something to be present to see if photosynthesis was happening or not. Okay, so each chloroplast suspension was mixed with DPIP, an electron acceptor that changes from blue to clear when it's reduced. That means when it's gained an electron, right? So when do we know electrons start to show up? Well, they start to show up when photosynthesis starts, right? When that light energy breaks water and water donates an electron, correct? Okay. Each sample was placed individually in a spectrophotometer and percent transmittance was recorded. Okay. So percent transmittance is how much light's getting through, right? So if a lot of light's getting through, that means it's not blue, it's clear. And if a little bit of light's getting through, that means it's mostly blue. Like the light can't get through it, right? Meaning no electrons are being accepted. Okay. The differences in the curves of the graph data indicate that there were differences in the number of electrons produced in the three samples during the experiment. So first of all, it wants you to discuss how electrons are generated in photosynthesis and why the three samples gave different transmittance results. Okay, so first of all, let's take a look over at photosynthesis. So first thing that happens in photosynthesis is we have the light reaction, correct? In the light reaction, light hits photosystem 2. Remember, these are out of order, right? And energizes those chlorophyll molecules, okay? And then what donates an electron? Water. And then those electrons become excited and they move along the electron pathway to photosystem 1, where they're rezapped again, and the electrons move even further. Well, what's generated in the process? We have some ATP that's generated in between the two of these, right, from that powering. And then in that second zap, we have ADPH that's generated, right? So we go from water donating electrons, moving down that electron chain, and then we have ATP and NADPH generated. So going back to our question was, discuss how electrons are generated. Okay, so we wanted to talk about, first of all, that the chlorophyll system is the link between the light and the generation of electrons. So those photosystems, photosystem 1 or photosystem 2, getting zapped. Where did the electrons come from? Water. That electron generation is not simply photosynthesis. It is proportional to the dPIP reduction in light transmittance. Okay, so it told us in the question that as dPIP goes from blue to clear, it's accepting electrons. Well, where are the electrons coming from? Photosynthesis. So how is transmittance tied to this? Okay, so transmittance says that if we have a chloroplast suspension that's not wrapped in foil with dPIP, we should have a high level of transmittance because this is going to be clear, going completely to clear from blue, right? Because we have lots of electrons being generated from photosynthesis happening. Okay. Next, chloroplast suspension surrounded by foil to provide a dark environment. This is going to have a very low transmittance, right? Very low transmittance because it is still mostly blue. Not a lot of electrons have been donated or reduced, right? So it's not turning to clear. And then the third one, the chloroplast suspension that has been boiled. Well, is this one going to generate electrons? Well, it's not going to generate electrons because of the denaturing of that enzyme, right? The photosystems here are types of proteins, right? Photosystem 2 is a protein. Photosystem 1 is a protein. If we boil that, these cannot work anymore and can't accept electrons from water, right? So in that second container, we had no light coming in, right? That was the stopping point. In our third container, we had plenty of light. We had just boiled the enzyme. So our solution's still going to be blue. So a lot of you did fine with the first part here, water donating electrons, photosystem 1, photosystem 2. Not a lot of people hit this, right? The electron generation is proportional to the dPIP reduction light transmittance, meaning it's not the turning of blue to clear as it's accepting electrons, and then the transmissions being high with that clear solution. Okay. Another thing, decreasing light availability decreases the quantity of electrons that will be generated. Right? That was another thing you could have talked about. A lot of people got that the boiling is going to denature the photosystems. Okay, so they couldn't just say it was the chloroplast was denatured or denaturing of a protein, a membrane's not working. You had to actually say that it was photosystem 1 or photosystem 2. So this was a huge 6-point question. You had to know what was going on in the experiment, and you had to know about this, right? Water donating electrons, and then the light powering them as they move through that chain there. All right, so next is question 3. Question 3 says, describe the most likely effects on cellular process. Be specific. Sorry, I'll go back to the top. In a certain prairie community, a dominant prairie grass species has recently been infected with a virus that disrupts one of the electron transport proteins of the chloroplast in the infected cells. One of these isn't working, right? Photosystem 1 or photosystem 2, okay? So describe the most likely effect on the cellular processes. Be specific as to which process and molecules are likely to be directly affected. Well, our light reaction is directly affected, right? If these are killed off by a virus, water can't donate electrons anymore. We cannot generate ATP, or not we, the chloroplast cannot generate ATP from the light reaction, and it also cannot generate NADPH, right? Without those two, what's the next cycle that happens? We have, where are these guys headed to? ATP and NADPH are headed out to the Calvin cycle. So without them, the Calvin cycle can't happen. What are we not going to generate? What's generated by the Calvin cycle? We're not going to generate ATP. Okay, we're not going to generate glucose, right? So glucose would not be generated in this process. Right? Okay, so as we big component here with the glucose missing. Okay, so points that could have been earned for this one, less ATP produced. There we go, right? So going back to this one, less ATP produced if these guys aren't working. What's another one? Less NADPH. There it is right there. Less of this if these proteins are not working, right? Very good. Inability to fix carbon. So remember we talked about how this cycle is called carbon fixation. So without ATP and NADPH, carbon cannot be fixed into glucose, right? Another one, a decrease in oxygen production. And eventually we know that oxygen is a byproduct of photosynthesis. So oxygen production is going to be less as well. So a lot of people started going into respiration. I'm not quite sure where that was coming from, but plants do respiration. Yes, that is correct. But melting these membrane-bound proteins in our light reaction is not necessarily going to directly affect respiration. It will eventually because you're going to be running out of oxygen produced for the plant, right? But these are our key things that are happening at first. Now, another thing I want to touch on, when you guys are answering your FRQ questions, I'm seeing some responses where it's this. Like I'm seeing this written word for word and not even really a complete sentence. This is giving you hints. Your details on how you're answering the question and showing me that you understand what's happening is the key. So please be specific. Please don't write exactly what's here. And I'm so glad you're going to these to use them, but please add some of your own information to this so it shows me that you understand what's going on. All right, guys, I hope that was helpful, and shoot me a message and remind if you need any further assistance.